Top 7 was a surprising breakthrough in protein design. This success led to a new idea that David Becker's team could use the software in reverse. Hello and welcome to a new episode of Wired and Weird, where we explore some of the most amazing scientific, technical and AI insights that are revolutionizing our world and touching our day-to-day lives. This episode we are talking about David Becker, who also shared the Nobel Prize for Chemistry 2024 for his achievements in mastering life's building blocks and creating entirely new proteins. This will have a deep impact in every aspect of our lives. After the Nobel Prize declaration of 2024, it is not the mission is going to replace chemists. It is that the chemists who use machines will replace those that don't. When David Becker started studying at Harvard University, he chose philosophy and social science. However, during a course in evolutionary biology, he came across the first edition of now classic textbook, Molecular Biology of the Cell. This led him to changing his direction in life. Eventually, he became fascinated by protein structure and he took an biochemist's great challenge to explore how proteins fold. At the end of 1990s, he began to develop computer software that could predict protein structure, which is called Rosetta. CAS competition in 1998 using Rosetta, and in comparison to other participants, it did really well. This success led to a new idea that David Becker's team could use the software in reverse. Instead of entering amino acid sequence in Rosetta and getting protein structure out, they should be able to enter a desired protein structure and obtain suggestions for its amino acid sequence, which would allow them to create entirely new proteins. Protein design, which involves creating custom protein with new functions, gained momentum in the late 1990s. Researchers often modified existing protein to break down hazardous substance or serve as a tool in chemical manufacturing. The field of protein design, where researchers create bespoke protein with new functions, began to take off at the end of the 90s. In many cases, researchers tweaked existing proteins so that they could do things like breaking down hazardous substances or functioning as a tool in the chemical manufacturing industry. Designing brand new proteins from scratch, so-called de novo design. Researchers sketched a new protein structure and used Rosetta to figure out the needed amino acid sequence. Rosetta searched a database of known protein structure for similar fragments, optimized them and suggested the best amino acid sequence. Baker's team tested the software by inserting the gene for the amino acid sequence into a bacteria, which then made the protein. They confirmed the protein structure using X-ray crystallography. Rosetta successfully created proteins named Top 7, matched with the researcher's design structure almost perfectly. Top 7 was a surprising breakthrough in protein design. Unlike previous de novo proteins, it had a unique structure not found in nature and was larger too, with 93 amino acids. After confirming AlphaFold 2 worked Dennis Hamis and John Jumber, who shared Nobel Prize with David Becker, calculated the structure of all human proteins and predicted the structure of nearly all 200 million proteins discovered in Earth's organisms. Google's DeepMind made AlphaFold 2's code public, turning into a valuable tool for researchers. By October 2024, over 2 million people from 190 countries had used it. While protein structures once took years to determine, AlphaFold 2 does it in minutes and estimate the accuracy of its predictions. Baker also released the code for Rosetta. So a global research community has continued to develop the software, finding new areas of application. After the 2020 CASP competition, David Baker saw a potential of AI models and added a transformer-based AI to Rosetta, significantly aiding in the de novo design proteins. This highlights the crucial role of AI in advancing protein design. RosettaFold is an AI-based 3-track neural network that predicts protein structures and build models of complex biological assemblies. It used deep learning and generative diffusion-based architecture to produce 3-dimensional protein structure quickly and accurately. Based on limited information, without the aid of such software, it can take years of laboratory work to determine the structure of just one protein. It considers patterns in protein sequence, interactions among amino acid, and potential 3D structure. RosettaFold can predict static protein structures, protein-protein interaction, and interaction for protein, DNA, and RNA. This ability to create proteins that are loaded with a new function is just as astounding. This can lead to a new nanomaterials, targeted pharmaceuticals, more rapid development of vaccines, minimal sensors, and greener chemical industry. The simplest ongoing example is the work of scientists trying to find ways to break down the 150 million tons of soda bottles found in the ocean, in the waste dumps, and in your backyard. The key would be use this protein database to examine the 3D shape of the certain proteins, the enzymes capable of splitting apart molecules of plastic and rendering them harmless. This may also have immediate medical applications because a number of incurable diseases are linked to misfold protein prions, which are linked to a host of incurable diseases that can affect elderly, such as Alzheimer's, Parkinson's, and ALS. AI and quantum computing are illuminating the unknown quantum world, lighting the way for humanity's future. With de novo design, we are catering entirely new proteins, making the impossible possible. Thank you for watching. Stay tuned for more insights on how AI is shaping the future of our nation's wealth and prosperity, and learn how you can stay ahead in the rapid involvement landscape. Don't forget to subscribe, like, and watch our next video for a deeper dive into the exciting future of AI. Thank you.